We used a genetic mouse model of Alzheimer's disease that develops age-dependent accumulation of vascular amyloid to see if the amyloid cast disrupting blood flow does play a role in the progression of the disease.
Alzheimer's disease is commonly associated with a reduced cerebral blood flow to the cortex. This means that the damages inflicted onto the nervous system due to this may contribute to the initiation and progression of the disease. Alzheimer's disease causes the anatomy of the gliovascular unit to be altered. Since vascular amyloidosis is a pathological trademark of the disease, we hypothesize that vascular amyloid can interfere with gliovascular signaling, which then contributes to cerebral hypoperfusion.
Amyloid beta comes from a large protein found in the fatty membrane surrounding nerve cells, and these protein deposits are a pathological signature of Alzheimer's disease, and amyloidosis usually precedes cognitive decline by several years. Amyloidosis comprises a lot of pathological conditions that occur after an abnormal protein called amyloid is deposited into various tissues and organs. Amyloid beta is toxic to neurons, and it also negatively impacts the vasculature by narrowing the lumen of vessels, causing vasoconstriction and forming plaques around blood vessels to disrupt the function of the gliovascular unit. In normal neural physiology, glial, neural, and vascular cells work together to form the gliovascular unit to ensure a blood supply that is correlational to the amount of neuronal activity occurring. The unit does this by constantly adjusting the cerebral blood flow to meet the fluctuating energy demands of the CNS, a process called functional hyperemia. Astrocytes can be found within the gliovascular unit, or GVU, to mediate this process, and they do this via large perivascular end feet that cover the entire vasculature. Astrocytes respond to neural activity by modulating intercellular concentration of calcium, causing the release of vasoactive factors that then affect the contractile cells in the GVU, the vascular smooth muscle cells, or VSMCs, as well as pericytes to change vessel diameter.
Amyloid deposits in the GVU are produced in mice with the use of HAPPJ20 in an Alzheimer's mouse model. We use coronal non-transgenic and transgenic mice brain slices to display different anatomical GVU arrangements in the transgenic mice. We incorporated immunohistochemistry to label the vascular amyloid astrocytes and vessel walls, which were observed under HD electron and multiple photon microscopy imaging. Interaction between the regulation of the vessel diameter and beta amyloid were observed by chemically stimulating VSMCs and astrocyte in the GVU. The vasodilation of the vessels were stimulated using norepinephrine TACPD. In contrast, ET1 and U46619 were used as vasoconstrictors. Changes in vessel diameter were observed through a 3D electron microscope. Then, in response to the vasodilation, and ex vivo slices were elected via calcium uncaging using laser pulses. The amyloid were marked green to ensure its location in the transgenic mice. Multiple photon microscopy through a chronic cranial window of the mice were also used to create HD in vivo imaging of the stimulated vessel.
To visualize vascular amyloid patterns in the brains of HAPPJ20 mice, we injected methoxyXO4 before in vivo imaging by multiphoton microscopy. Multiphoton optical slice data were captured through a chronic cranial window in the mice, using tritsidextrin to label the lumen of the vasculature. Our results indicate a close relationship between amyloid beta and the vasculature in the HAPPJ20 mice. 3D reconstructions, as shown in Figure B, depict how the bands of amyloid can span the entire circumference of vessels in some areas, not others, as shown in Figure C. In other Alzheimer's disease models, vascular amyloid forms a banding pattern between established VSMCs, and we also observe this in the HAPPJ20 mice. In Figure D, by using high-resolution confocal Z projections, we see the vascular amyloid had a banding pattern resembling an exoskeleton around blood vessels. Figure E shows amyloid bands forming bridges instead of rings, indicating that they have reached the point of no return. In Figure G, a quantitative assessment shows that amyloid volume was 20% of total vessel volume. Overall, we conclude that vascular amyloid appears to have formed a vascular exoskeleton covering a significant surface area of cerebral blood vessels in aged HAPPJ20 mice.
Next, we wanted to see if vascular amyloid deposits interfere with astrocyte and feet placement, so we imaged a cube brain slices from a mouse model using confocal imaging. Then, to label vascular amyloid, we injected methoxy XO4 followed by bath application with Lexo-463 hydroxide dye to stain the elastin layer of the vessels. Rod 2EM was used to label the end feet. Results shown that amyloid deposits did displace the end feet. Figure B shows that the end feet no longer but the elastin layer in areas where vascular amyloid were present. However, there's a few areas where the end feet did not get displaced, as shown in Figure C. Since Rod 2AM doesn't label all astrocytes, we used a water channel called AQ4, which is exclusively expressed in astrocytic end feet, to see if the end feet were placed in the proper position. Figure A depicts the GVU with astrocyte end feet expressed in AQ4. From the results, we conclude that the astrocyte polarity has been altered in our HAPPJ20 mice due to vascular amyloid disrupting the intimate end feet vessel relationship.
From the study, it was concluded that astrocytes respond to the vasoactive molecules TACPD and norepinephrine with increased calcium, which, depending on the oxygen tension in the tissue, cause either vasodilation or vasoconstriction. Thus, we use video microscopy analysis to measure changes in arterial diameter in response to bath application of TACPD or norepinephrine in acute slices from control and HAPP J20 mice, as shown in figures A and B. Most arterioles and control slices dilated in response to TACPD, and a paired analysis indicated a highly significant effect of TACPD, as shown in figures A and C. However, in HAPP J20 vessels, there was no significant effect of TACPD in a paired analysis, which means that dilation of vessels have been impaired in HAPP J20 mice, as shown in figure E. A similar effect was observed with norepinephrine, which induced significant vasoconstriction in control vessels, as shown in figures B, F, and H. To visualize vascular amyloid, we injected methoxyXO4 12 hours prior to control in HAPP-J20 mice. Then we used confocal imaging to identify vessels with and without vascular amyloid. We then determined changes in vessel diameter by using elexifluoride 633 hydrazide. Furthermore, to avoid possible off-target effects of the TACPD and norepinephrine that raise calcium in astrocytes, we elevated calcium in individual astrocytes using DMNP4 in combination with the calcium sensor rod 2AM. Focal laser uncaging allowed us to encage calcium in single astrocytes on or close to arterioles and cortical gray matter arterioles and slices from control mice, and arterioles with and without amyloid in HAPP J20 mice were imaged. In control slices, calcium encaging in astrocytes led to an immediate rise in calcium concentration followed by vasoconstriction, which is shown in. Now in slices from HAPP-J20 mice to determine if the impaired vasoconstriction was really due to the amyloid burden on the vasculature, we compare the vasoconstriction of arterioles with and without vascular amyloid. In the HAPP-J20 mice, the response of arterioles without amyloid was similar to arterioles from control mice. In contrast, the presence of vascular amyloid significantly decreased vasoconstriction, as shown in the right animation. The data that we captured then suggested that arterioles burdened by vascular amyloid do exhibit hindered vasoconstriction.
We deduce that reduction of vessel contraction after astrocyte stimulation could also occur due to impaired VSMC parasite function. To assess this directly, we use two potent vasoconstrictors that bypass the astrocyte component of the GVU, ED1 and U46619, and stimulated VSMC's parasites directly instead. Then we measure change in arterial diameter in response to bath application of ET1 and U46619. Arterioles from HAPPJ20 mice had significantly less vasoconstriction in response to ET1 and U46619, as shown in figure B, and compared to control mice, as shown in figure A. Taken together, these data suggest the ability of VSMC parasites to properly function and regulating vessel diameter is impaired in HAPPJ20 mice. The graphs on the right are graphical representations of figures A and B to further assess if a greater initial vessel tone can have led to decrease in vessel responses. We compared baseline diameters of both capillaries and arterioles. The graph on the left shows that neither baseline capillary or arterial diameters were significantly changed in HAPPJ20 mice, which allowed us to conclude that the reduced constriction of vessels seen in HAPPJ20 mice could not have been due to a difference in baseline diameter.
We next examine potential changes in VSMC function in vivo. We viewed the vasculature and amyloid through a chronic cranial window in live mice, and we used direct focal optical stimulation of VSMCs to induce vascular response. We then compared the vasoconstriction of arterioles with and without vascular amyloid in HAPPJ20 mice, and we saw that arterioles without amyloid constricted right after activation of VSMCs, as shown in figures A and E. Meanwhile, arterioles with amyloid had reduced constriction, as shown in figures B and E. Then we observed vessel areas with and without amyloid on the same arteriole to see if the impaired vasoconstriction was restricted to vessel areas with vascular amyloid, and we showed that areas with amyloid constricted less than areas just downstream, as shown in figure C. These in vivo data are similar to our ex vivo data, which then led us to conclude that arterioles definitely had a reduced range of constriction, specifically in areas covered in vascular amyloid.
A future study could involve AQ1, another water channel that is expressed in the entire astrocytic surface, so we can conduct another experiment to see if engaged AQ1 expression can compensate for AQ4 loss. A clinical application of our study is that the data show that there's a certain type window for therapy during which amyloid clearance will allow the vessel to regain its ability to constrict, thus the impairment due to the GVU could potentially be reversed.
Some critiques of our experiment are that our model system was not perfect since the mouse brain is not the same as a human brain. However, it was still a good model because it was a transgenic mouse model that produced the human version of the amyloid protein and recapitulated a lot of the pathology. Disease progresses faster in the mouse model and thus kills it